So, hello everyone. Thank you for the organizing team to have given me the opportunity to present my PhD, part of my PhD work that I conducted with Sonia Pellissier, Pascalotte, and Astrid Kieber at the l'Université Savoie-Mont-Blanc in the Alps in France. And today I'm going to talk about how stomach and brain can interact together in terms of oscillatory coupling, and how its dynamic are influenced and co-influenced by psychological traits. So as we all know here, gut-brain axis plays a key role and is also influenced by affective traits and anxiety levels, stress, but also we saw this morning that it could be related to cognitive functions such as decision-making. But here and now, I'm going to focus more on among all these interactions that can appear between the gut and the brain. I'm going to focus on the oscillatory electrical oscillatory coupling between the stomach and the brain. And especially, I'm going to focus on the electrical oscillatory rhythm, which is interestingly generated by the stomach, inside the stomach wall, by the interstitial cell of Cajard. And this oscillatory rhythm also the muscular contraction and the activity of the stomach. And so from this wonderful paper of Ignacio Ribolo, we can see here that the electrical signature of the stomach is around 3 cycles per minute in healthy human, with a 20-second cycle of 20 seconds long of duration. And we can measure or take a glimpse of this oscillatory compound using electrogastrography, which is a non-invasive measure. We basically put some electrode on the belly on the abdomen of the participant and record this electrical activity. So from this oscillatory activity, we can extract the phase. And what kind of interaction we can extract is the interaction between the phase of the oscillatory rhythm of the stomach and the ongoing amplitude of the power of the oscillatory rhythm of the brain, which can be measured, and as we measured here, with electroencephalography. So we are talking about phase amplitude coupling here between the phase of the stomach rhythm and the amplitude of the EEG power. And so that's what, again, the same team did as a pioneering work, amazing work from Richter and colleague, which showed for the first time that the alpha power was coupled to the rhythm of the stomach at rest in healthy subject and in a more ascending manner. So from mostly stomach to brain, even if we cannot exclude the other way around. But at this stage, we didn't know really what was the functional role of this coupling, but considering the region, so anterior insula, but also the occipital cortex, so we guessed, or we hypothesized that this stomach-brain coupling could be related to interoceptive functioning and emotional regulation processes, and also cognitive processes, which involve exteroceptive processing, the exteroceptive processing of exteroceptive stimuli. Sorry. So, but now from what we saw in this morning, the occipital cortex is much more related to other kind of stuff than only visual stimulation. But we conducted a pilot study that we published in 2022, where we demonstrated in a virtual reality that the intensity of this stomach-brain coupling in alpha and frontocentral electrode was higher during a cognitive demanding task that you can see on the left, compared to a relaxing task using a cardiac biofeedback procedure. But now I'm going to present some results where we tried to extend these results to other cortical oscillation and other scalp regions. And also we wonder if this stomach-brain coupling and differences were sensitive, were sensitive to disorder of the gut-brain axis. And so we try to look at what would happen with irritable bowel syndrome, which is a multifactorial gastrointestinal disorder, whose symptoms include gastrointestinal pain, gastrointestinal motility issue, but we do not have actually clinical organic index of this disturbances. And it is also, they have the symptoms that are increased by stress and anxiety levels. And so we can find also comorbidities such as anxiety, neuroticism, and visceral hypersensitivity. But what is hypothesized is that there is a neurovisceral integration default, so a disturbance into the gut-brain interaction. So we wonder if the stomach-brain coupling dynamics in irritable bowel syndrome was similar or different than those of healthy subjects. So to do so, we perform an analysis with healthy controls and irritable bowel patient matched in age and gender. We recorded multiple electrophysiological measure, but I'm going to here focus on the EEG and EGG. If you have question on the electrodermal activity, sorry for the typo. I will answer it, but I will not have the time to present it today. And we measure the interoceptive awareness as a measure of the global trait of the global ability of people to sense, to be sensitive to their interoceptive feeling, their body feeling. And we measure also the trait anxiety and neuroticism as a measure of tendency to feel negative emotion. So we performed 2 sessions, 24 days spaced, and during these sessions, the participants underwent 30-minute resting state, and then they perform a cognitive demanding task, and then we measured also their activity during a recovery period of 30 minutes. And the cognitive demanding task is a modified stroke task with an increasing level of difficulty, which induce also upward social comparison, and we also perform a double task in the end to saturate their cognitive resources. And so what we found is that in terms of stomach-brain coupling intensity, we found that there is a strong module effect. So the stomach-brain coupling changes between the rest, task, and recovery period, and mostly and most significantly between 10 and 13 hertz. So again, in the alpha power. For the sake of simplicity for this presentation, I'm only going to focus on what we analyzed in this bandwidth. And so what you can see here for healthy control, so healthy subject, is that the more the topographic map is red, the higher the stomach-brain coupling intensity. And so you can see that there is a huge decrease in almost all of the scalp between the rest and the task, and then it slowly recovers during the recovery period. But in frontocentral, on the left frontocentral electrode, this effect was marginally significant. And contrary to our hypothesis, we have the same behavior in irritable bowel syndrome, and it was anecdotally confirmed using a Bayesian analysis. So then we next wonder if the psychological trait, so anxiety, neuroticism, and interoceptive awareness could explain the inter-individual variability in terms of dynamic of stomach-brain coupling along this task. So we model with a quadratic polynomial function the intensity of stomach-brain coupling through time, as you can see on the right as an example. And we perform a partial risk correlation analysis in healthy subject and in irritable bowel syndrome patient. And we tried to see if these traits were related to these dynamics and differently related to these dynamics in irritable bowel syndrome. So what we found in healthy control is that the higher the neuroticism and anxiety trait, and the lower the interoceptive awareness, the stronger the change in stomach-brain coupling dynamics. So as you can see here, in the central electrodes. And interestingly, when we applied exactly the same weights that we extracted from this analysis to irritable bowel syndrome, we found the reversed, significantly reversed relationship. So stomach-brain coupling phase amplitude dynamics seems to be a meaningful index to quantify psychophysiological regulation, and it is mostly disrupted in alpha power, and it slowly recovers. So the timing of this interaction needs to be clearly taken into account. And we are so it's only a question, but we wonder if it is a reflection of a disembodiment during the task center to the stomach that we could observe with this reduction of stomach-brain coupling. Or is it synchronization of brain activity towards more exteroceptive cue and exteroceptive task instead of interoceptive one? And the reverse association that we found between psychological trait and stomach-brain coupling dynamic between the two populations, we wonder if it reflects a different type of cognitivo-emotional regulation strategies, and could reflect a way of being resilient to an event, stressful, or in this case, a cognitive demand. But I hope that I could have at least a bit convinced you that the stomach-brain coupling dynamics could be a key measure of the body-brain-mind axis, and help the modeling of functional gut-brain axis design. So thank you very much for your attention. Sorry for the little delay, and I will be happy to answer your question now. Yeah.
Thanks for the really nice presentation, insightful. I have a few questions, and you touched upon it yourself, and that is the question of directionality.
Yeah.
I mean, you have now a measure that clearly gives you insight that something is dynamically changing, but is it the gut driving the brain, or the brain driving the gut? And then you link this to your traits, and then you can start thinking, okay, what is the driver here?
Yeah, so the causality analysis is undergoing. I don't have the results yet. But from the knowledge that we currently have, so at rest, the alpha stomach-brain coupling is mostly ascending from stomach to brain. And this is what is disrupted in our design without any change in, that I didn't show you that, but without any change in the pace setter rhythmicity or amplitude. It's very stable through time. So my guess, even if a causality analysis is needed, is that is more disruption of ascending processing by the brain, a stop of the cortical synchronization dynamic with the stomach. So, like as it begins to be said, maybe the stomach act as a metronomus, metronomus of the cortical organization. But
since you have such beautiful data over time, have you also considered to actually do a brain state analysis of your data to actually look exactly at how people, well, in a sense, experience that dynamic while they undergo a task, for example?
Yeah. But I didn't have the time I didn't have the time at the beginning to do that. But yeah, okay. I consider doing that. Yeah. Maybe in the next study. Yeah, you're welcome.
Question relates to this question here. The stomach obviously has different motility behaviors, right? I mean, the stomach can do peristalsis, it can relax, it can
Yeah.
And all these are reflected in different activities of those cells that you showed in your introduction. So they will affect your measurements. I was wondering in which motility statement or basis these patients were. Because that's gonna give a huge effect, of course, on the activity.
So
It's probably the effects that you see in your ascending effects on the brain.
So yeah, basically the study was we always perform the study at the same time of hour, and we asked for a controlled meal. So we checked after. Of course, we cannot. We just ask them to a typical meal to eat before.
But of course, there's a lot of variability between people. One person might start digesting immediately, the other one. I mean,
yeah, of course, we started the recording at least 2 hours after this meal period, and we checked that they effectively took a meal, but by asking them to count their explain what they did the morning.
yeah, but you don't have an idea about which mobility pattern was ongoing.
No. Of course, the electrogastrogram is a combination of muscular contraction, but not always, and rhythmicity. What the indice in electrogastrogram, which is more correlated to muscular contraction, is the amplitude. We did not have any change in terms of amplitude. And I didn't show that, but we controlled for this relationship for pace setter amplitude, pace setter rhythmicity, and none of them predicted what we found on the blood-brain axis. So
then my next question is, you study IBS. Obviously, IBS is more a syndrome of the lower gastrointestinal tract, like large intestine. Wouldn't it be more appropriate to look into something like functional dyspepsia, where the symptoms arise more from the stomach and the upper small intestine?
Yeah, of course. But like the stomach is also the first part of the gastrointestinal tract, and so what happens here has a reflection in that. Of course, maybe what we would have loved is to measure the intestinal activity, which we tried, but it's much more harder to extract a pattern that is reliable compared to the stomach activity. So we started on studying the stomach activity with the assumption that what happens in the stomach can have a repercussion on the gastrointestinal tract. Yeah.
Small question.
I don't know.
Right. Sure. Yeah. Sure. So can you say nice thought? Can you say that the stomach's brain dynamics is that also a comparable measure for the vagus nerve activation? Do you think is it a more direct reflection of the vagus nerve, by which seems to be more logic that neuroticism?
Yes. Yeah, so for the sake of the time, I didn't present all the data, but we tested. So we tested if the parasympathetic system and sympathetic activity was correlated to these changes in terms of stomach-brain coupling intensity. And surprisingly for us, we were guessing that parasympathetic activity would mainly predict this, but it was not the case. It was sympathetic activity. Yeah, so that's why we are more prone to say that it is like a kind of arousal activity that stop the cortex to consider in certain region to monitor the stomach rhythm. But yeah. I think that maybe this index, but for sure, sorry, but for sure, further research is needed. Much more further research is needed, but maybe it could be a better index of the vagal activity, but really the branch which innervates the stomach instead of what we currently do with HRV, which is linked to the branch which innervate the heart.
Thank you very much again.